Good afternoon, everyone. Hope you're enjoying the conference, and I'd like to thank the organisers for pulling this conference together. I've absolutely loved it so far. I'm delighted to speak today about a side project I'm working on, which is, sorry, is that something? No, which I'm working on as part of my PhD, and share some very preliminary data. Not only am I working on the side project to gear myself up for my actual project, which will be working on slow loris genomes, but also to produce research that I hope will help myself and other people later down the line. Right, there we go. Pardon the green box. That won't happen again, hopefully. Venom is arguably the finest evolutionary trait to study in animals. It's found in over 250,000 species. It has evolved at least 101 times. And perhaps most interestingly about the trait, it has at least 11 separate functions identified across the animal kingdom. And it's really been within the last 2 decades that we have truly entered a golden age of understanding the evolution of venom. Research has indicated that the evolution of venom is not simply a just so story of duplication followed by neo and subfunctionalization, but rather a much more complex, multifaceted process that differs depending on each species. That toxins are being recruited and then diversifying or purifying at a rate that is impacted by different traits, such as the age of the evolutionary ages of these groups, the life histories, or even the species richness. And that's something that I find particularly quite tantalizing, is that the evolution of venom and how the convergent evolution of venom may run much deeper than we really predicted. Well, there has been a lot of evolutionary work done on the more popular groups, such as snakes and cnidarians, for example. One group that gets consistently quite overlooked are the mammals. Mammals are the smallest tetrapod group that we have, approximately 6,500, I think, at last count. So not too many of them, but despite that, they still possess an enormous amount of morphological and species diversity, in part thanks to their adaptive radiation. Looking at the venomous mammals on the screen here, you can already see there's a great selection of traits that have made mammals so successful at exploiting countless niches. Considering this, I have to say, I don't think it's entirely surprising that venom has been selected for in the mammals. What is notable, however, is the distribution of venom across the entire class. It is present in 4 of the 20 different orders. That each of the orders have their own unique venom delivery system that doesn't, there's no shared factor, it's all very distinct across them. And that despite venom being only found in a very small handful of species, it has at least 6 functional uses for the mammals, which is quite a big deal. And again, especially when you compare these features with other groups, other venomous groups, beg your pardon, the mammals do quite stick out as a novel group worth a lot more investigation and research. Now, I'm not the first, this isn't like a brand new opinion, this isn't anything hot, this is something that has been thought about for ages, and there are absolutely fantastic already on venomous mammals. In fact, the previous 2 speakers and my supervisor, Professor Anne Macaras, they have produced the 3 most recent review papers out there. These reviews are fascinating because they use particularly ecological, behavioral, and proteomic data, as well as fossil data too, which is very interesting, to produce these reviews and really examine the evolution of venom within mammals. But within the last few years, and in particular this year alone, we have really started to see some fantastic genomic and genetic data on venomous mammals. So within the last year, as I just said, we now have the transcriptome of the short-tailed shrew. We have a brand new platypus genome, which goes to chromosome level, may I add, which is absolutely phenomenal. And again, there is evidence that oral venom systems, so such as those in the shrews and the solenodons, could be ancient conserved parallel evolution, which is quite a big deal. It's with this in mind, this fantastic foundation of brilliant reviews done before and the new cutting edge research, that I am aiming to produce a review examining the evolution of venom within mammals from a genetic and bioinformatic perspective. I've got 2 very preliminary aims, which is to try and identify patterns. Are we seeing any toxins that are perhaps more predisposed to show up within mammal venoms particularly, and also to compare and contrast toxic and non-toxic genes. So The most common one and mentioned already are callicrines, for example. You can find them in shrew and selendon venoms, but in other species that, like ourselves, they're not venomous. So is there any way we can is there any indication from the data of why that might have been happening? So to begin, I have extensively searched through the literature for any proteins or toxins that are implicated within mammal venoms, and also have nucleotide and proteomic data. I think that's very important. I want to stress that because I'm sure you will know there are a lot more than these guys out there, but I need that genetic data. So this is what I've got. This is a summary of the table that I've been collating of nucleotide and protein sequences. And currently there are about 204 separate entries representing 5 species and all 4 orders. For the purpose of today's talk, I'm just going to be focusing on some of the results I have from nucleotide vampire bat sequences. Vampire bat does represent the bulk of my data, so it felt like a good place to start. Also, I started in January, so I haven't gotten too far, if I'm being very honest with you. Current pipeline as it stands is, first of all, to download these sequences, next to blast against mammal-specific related databases using the maximum amount of hits possible. Then we are filtering the hits based on the e-value and the coverage of the query sequence as criteria. Just to briefly explain for everyone, when I say e-value, I mean how likely, the closer it is to 0, the how likely it is an actual hit against your query sequence and displays some level of very worthwhile homology, as opposed to a bunch of random nucleotides that just happen to, match with the end of your sequence. Finally, I've then aligned these sequences, and then the data I'm showing you today is made using maximum likelihood, which is an absolutely marvelous phylogenetic technique. What I've done is I have highlighted the vampire bat sequences with a funky color and a bat icon, because we love visual representation, folks. So first, I'd like to show you lactorin, which is a protein that promotes secretion. Those of you familiar with scientific names might already notice something quite peculiar, which is that there are equid and feliform carnivores being grouped together with our lactorin, with the vampire bat lactorin hits here. Particularly, you have Przewalski's horse and notably a lot of the larger cats. As spoken about today and yesterday, we know that phylogeny, does not necessarily reflect the genome or phenotype of venoms. And that's one of the aspects that, to me personally, I think makes this a fantastic area to research. And this recovery of horse and cat sequences with the bat is not brand new. This has been reported before, and in particular with a certain group, which are the desmopilions. Again, pardon my pronunciation on some of these proteins, which is a salivary androgen protein that it was identified in Dracula's Children, a 2013 paper by Lowe et al., that it is a derived form of the cat and horse allergen, and is also acting under quite high diversifying selection. So what you can see here is I've highlighted the vampire bat sequences. There's quite a lot of them. That's nestled within Chiroptera with other bats, but that box I've highlighted up there shows something very interesting. So not only do we see equid, but for the first time, as far as I'm aware, we also have white rhino and Southeast Asian pangolins reported in the mix as having quite similar sequences. As you've seen from the previous talk, these sort of, what's the word I'm looking for? Beg your pardon. It throws up this very interesting question of the patterns, the types of toxins that we're seeing repeatedly occurring throughout venoms on a broad level, but also it talks about how quite an interesting level of homology as well. Why are we getting these very unusual, to go against the previous speaker, pardon me, non-toxic sequences when we compare them with what we know are actually toxin sequences? And it's going on this point of patterns that I just finally would like to show you some kunits, which I'm sure all of us here as venom scientists are at least somewhat familiar with. Good old kunits is always showing up in venoms. What we have here, again, vampire bats, and even more interestingly, Sorex araneus with the platypus as well, being grouped quite closely to the vampire bat sequences. So again, bringing up this question of convergence, these reoccurring patterns. However, as I said, this is all very preliminary data. I've still got an awful lot of work to do, and there's definitely a lot more that I want to be doing on this work. But already to see some very peculiar results is absolutely thrilling, because it really does crack open this question of how has this trait evolved, and is there anything really strange about it? So future work, very briefly. Number one, going to be doing further phylogenetic analysis on all the other proteins and nucleotides. So I'm going to be doing maximum likelihood and Bayesian. I think it's very important if you do phylogenetics that you do both. That's just my hot take. And finally, I also want to go further beyond the phylogenetics and use some population genetic techniques, possibly a bit of networking, dare I say it as well, to really pull apart and squeeze as much juice out of this data and really have a look at what's going on in there. Do have a few acknowledgements. I would like to acknowledge both of my wonderful supervisors, Dr. Ravinda Carpenter and Professor Anna Nakaris. I have really appreciated all the support they've given me, especially theoretically and technically. I am really looking forward to the next 3 years, and they know that they're stuck with me for that. So let's see how it goes. I also want to thank many of my fellow PhD students in the labs that they are part of, because they have been such a support network, and I'm very grateful for them. Even just to talk to someone who's like, yeah, I get exactly how you're feeling, downloading 3,000 sequences of a cow, for example, hasn't happened yet. And I also know it's a bit cheesy, but I would like to thank my family, who up until June I was living with, and they were there through the bulk of my collection and my downloading the database, and God love them. I used to come downstairs and be like, someone said the mole is venomous again, and now I need to see if I can find any sequences about it, and they would just rock and roll with it. To have a group of people who so unfailingly support you is absolute joy. I want to make them and all these people really proud. Thank you so much for listening.